And my name is Neta Zuckerman. I'm a computational immunologist in practice. I'm a postdoc. I work at City of Hope, which is a cancer research hospital in LA, and both here in collaboration with JU from Andrew Goldschmidt's lab. So let's start. So before we start diving into the material, first I really like to define the term biology. So what exactly is biology? So biology is defined as the scientific study of living things. Living things are also called organisms. And when we say living things, what exactly characterizes something that is living? So what is the criteria for something to be alive, to be considered as alive? So here are the criteria for all living things. And first of all, the first criteria is that the smallest unit of every living thing is a cell. And so every living thing has to be composed of cells. And some living things are composed of one cell. They're called single cellular organisms or single cellular living things. And a lot of other living things are multicellular organisms such as ourselves, such as humans. And so why are organisms called organisms? Because they are also highly ordered. So if you take a bunch of cells and put them together, you get a tissue. And if you get a bunch of tissues and put them together, you get an organ. And a bunch of organs put together actually make up an organism. So that is the origin of the name. And so also every living thing has to be able to manipulate energy, which is basically what we refer to as eating. And in the process of manipulating energy, every living thing absorbs food or nutrients from the environment and manipulates them and metabolizes them to produce energy, which then moves throughout the organism to make it work. And we'll see later on that everything that we need to make work in the organism actually uses energy. Actually, also every living thing has to be able to reproduce. And by reproducing, I mean that every living thing has to have DNA, or the genetic material. We'll talk about it a lot later. And so every living thing actually inherits DNA from its parents and then passes them on to the offspring. Also, every living thing has to respond and adapt to the environment as part of getting used to the environment that they live in or part of survival, and every living thing needs to grow and develop. And so in biology, one can study many subjects and many things. One can study botany or zoology, molecular biology, which is everything on the molecular level, or virology, or learn about bacteria, but fortunately we do not have that much time. Oh, and I totally forgot about this figure. This was mentioned before. This is actually, so when we talk about organisms and living things, this is the tree of life, which describes all the organisms that are now known to exist on Earth. And it describes the relationships between them using a phylogenetic tree. And so you can see three main groups here. So the bacteria in archaea are actually single cellular organisms. They only have one cell, whereas the eukaryota are multicellular organisms. And if we're looking for us in this tree of life, we can see that there is one group called Animalia, and the Homo sapiens, or the humans, are under this group. And so the topics that we'll cover today are genetics, inheritance, and how is the DNA involved in genetic inheritance, and several processes that the DNA is involved in. And tomorrow we'll dive more into the world of proteins and signaling networks and give several examples about that. So any questions? Okay, so let's start. Oh, and throughout the presentation you'll see this slide. This is basically to give some order to the presentation. Sometimes with a lot of new terminology, and I'm going to give you a lot of new terms and a lot of new words that you might have not heard before. And just we can follow the different topics that I'll cover throughout the 2 days, throughout this presentation, and every time I'll go into a new topic, it'll be highlighted in red, such as now we're going to talk about genetics. So between the years of 1822 and 1884, there lived an Austrian monk called Gregor Mendel. And back back then he might have been known as an Austrian monk, but today he's very famous, and he's known as the father of modern genetics. And this is because what he did is he carefully studied the patterns of inheritance, simple patterns of inheritance in pea plants. And he carefully recorded everything that he found, and he gathered all of his findings into what is now known as the laws of Mendelian inheritance. And these basically describe the inheritance of traits in pea plants, which follow particular patterns. And until today, we can see some of these patterns, or the more simple patterns of inheritance, that are actually also seen in humans. And the reason that Mendel chose peas out of all different organisms that he could have chosen is because, first of all, they're very easy to grow, so there's relatively low, short amount of time since they're very small until they reach their full-grown size. They come in a variety of shapes, sizes, and colors that Mendel actually investigated. So these are the 7 characteristics that he has investigated, such as the shape of the seed, whether it's inflated or dented, the seed color, if it's yellow or green, etc. And most importantly, he could really control the reproduction of the pea plant really well. So under normal conditions, under situation, if nobody meddles with the pea, then the pea plant can actually self-fertilize. So it has both the male and the female organs, and the peas actually self-fertilize and are crossed with themselves. And what Mendel did is he basically meddled in this self-fertilization, and he actually took the pollens from one pea and brushed them on to another pea, while making sure that the anthers, which is the male parts, are removed from that pea, and therefore he could actually control which peas he wanted to cross with each other. So what did Mendel do exactly? So this is the first experiment that Mendel has made. And what he did in this experiment was he crossed between 2 types of peas. So he crossed between a pea with a spherical seed, so a round seed, and a pea plant with a dented seed. And these 2 pea plants are called the parental generation because they are the parents and they are about to be crossed. And most importantly, that they are true breeding. And what does true breeding mean? True breeding basically means that if you take the pea, the spherical seed pea, and you cross it with itself many, many times, you will always know the end result, because the end result will always be a spherical seed pea. And the same with the dented seed. So now when he knew all conditions, he could start with his experiment. So his experiment was to cross between these 2 peas. And when he crossed between these peas, he got this F1 generation, the first offspring generation, which is this generation that we see here. And what he saw that all F1 generation included spherical seed peas. And he noted to himself that the dented seed characteristics seemed to disappear within this F1 generation. What he next did is he crossed the F1 generation with itself to get the F2 generation. And the results that he got are very interesting. And also, no matter how many times he repeated the same experiment, he always got the same results, or very similar results. And what he got are always the same proportions. So he always got 3/4 of the pea plants that were spherical, and only 1/4 that was dented. And he also noticed that the dented characteristics seem to reappear in the F2 generation. And so what he defined next is that each characteristic, such as the characteristic that defines the shape of the seed, so the seed can either be spherical or can be dented, so that each such characteristic is defined by something that is called allele. And each individual, or each pea plant, possesses 2 alleles for each characteristic. So right now we're only focusing on one characteristic. We're focusing on the shape of the pea plant, and the shape is either spherical or dented. And so what controls the shape of the pea plants are 2 alleles. And why 2? Because 1, the pea plant receives from the father, and 1, the pea plant receives from the mother. And so you can see that the spherical seed, he knew that the 2 alleles for the spherical seed, parental generation, true breeding pea, was, and I annotated the spherical seed as large S in green, and the dented seed as small s in yellow. And he saw that for the parental generation, the 2 alleles, because they are true breeding, because we always know the result, we know that they will always get from the mother and from the father the same characteristic. Because when we cross them with themselves many times, we see that we get the spherical seed or this dented seed all the time. And so the 2 alleles determining the shape of the seed are large S in this case, and small s in the case of the dented seed. Now what happens when we cross these pea plants, like we did before? When we cross the seed plants, each parent provides seed donates one small s. And so we know that all the F1 generation look spherical, and their alleles are always large S and small s. One they get from the father, one they get from the mother. Now, the question is, what happens when we cross F1 generation with itself? So the F1 generation has two possibilities to give. It can either give the large s or the small s. And so to compute all the possible combinations of what the F1 generation can give, we use something that is called the Punnett Square. And the Punnett Square simply says, okay, what happens if the father donates a large s and the mother donates a large s? Then my F2 generation will have a spherical seed, and the alleles will be one large s, one large s, one large s from the father, one large s from the mother. When the father gives one small s and the mother gives one large s, then this is how the alleles will look. We'll have one large s, one small s, and the seed will look spherical. The same goes if the father gives one large s and the mother gives one small s, then we have the same case. However, if the father provides the small s and the mother also gives the small s, then all of a sudden our pea plant will have a dented seed, and it will have two small alleles, two dented seed alleles. And now we can also add another term, so we can see that if there is a presence of a large S allele in any of these cases, we always see the spherical seed characteristic, and that characteristic is actually called the dominant characteristic, because it only needs to have one allele for this characteristic in order that we can see that dominant characteristic. And the dented seed is recessive because we need the two alleles to have the recessive characteristic in order to see this recessive dented seed. Any questions? Okay, and so this was computed into Mendel's first law, and which basically states what we just said, with additional terminology, which we will soon see. And the first law is called the law of segregation, which states that each individual, each pea plant, has two alleles for each characteristic. For example, the characteristic that states the shape of the seed. And these alleles separate during gamete formation, meaning during the formation of the sperm and the egg. And then they reunite during fertilization, meaning the father donates one allele, and the mother donates one allele, and during fertilization, during the creation of the F1 generation, then I get this, the situation where I have two alleles again. And so Mendel came up with many other laws, and I'm not going to state all of them due to lack of time. However, these laws, some of them, so they are all true for very simple patterns of inheritance, such as we see here. And some of them are also true for laws of inheritance in human beings. And they are still used today. And so now we're going to talk a little bit about inheritance and what does it mean? What are these alleles exactly mean? What is an allele? So we haven't really defined it yet. So we all heard the word DNA. DNA is actually the genetic instructions or the book of instruction that exists in every living cell. And this is just a picture of a cell, and a cell has many organelles, many structures in it, and the DNA is actually kept inside the nucleus of a cell. It's a structure in the cell called the nucleus. And in every living cell, so if we have a multicellular organism, every living cell contains the same copy of this big book of instructions, this DNA, this genetic material. And so in the nucleus, the genetic material, the DNA, is arranged into shapes that are called chromosomes. So from now on, when I say chromosomes and when I say DNA, I actually mean the same thing, because we are used to think of DNA as just this long strand, and basically it's just packed into these structures called chromosomes. And this is the map of human chromosomes, and we can see that human chromosomes have 23 pairs of chromosomes. And this is the pair thing again. So why pairs? Why do we have pairs of chromosomes? Because one we get from the mother, and the other one we get from the father. Okay, just like the pea plant, one allele they got from the father, and one allele they got from the mother. And so each of us is actually composed of genetic material that we got from our parents. And when I say allele, what exactly do I mean by allele? So when I talk about allele, I actually talk about a little segment in the DNA that determines a certain characteristic. For example, in the pea plant, pea plants also have chromosomes. And the allele that determines the shape of the seed in a pea plant is actually a little segment here in one of the chromosomes, in one of these DNA sequences. And so the fact that humans, and also pea plants, by the way, have two pairs of chromosomes, one that they get from the mother and one that they get from the father, makes them a diploid. Makes them have 2 times n. N is the number of chromosomes. chromosomes. And N in the case of human beings equals 23. So humans, every single cell in human has 23 pairs of chromosomes, meaning 46 chromosomes. The pea plants, for pea plants, the N equals 7. So every pea plant, every cell in the pea plant has 2 times 7 chromosomes, meaning 14 chromosomes in each cell. A pair of chromosomes from the father and a pair from the mother. There are also haploid organisms or haploid living things out there, such as a lot of types of algae and fungi and fungus, and some types of male bees and ants are haploid and only have 1 pair of chromosomes. So when we say that these characteristics are inherited from the mother and from the father through the DNA, so we said an allele, a characteristic, a certain characteristic is just a part, a segment of the DNA. What do I mean by that? So let me introduce you to the circle of life. I'm sure all of you are familiar with it. And yeah, I chose to bring the example of humans and not pea plants because I think we can more relate to that. And so if we have 2 adults, and we already talked about that each cell in the adult is diploid. And what does diploid mean? It means 2. It means 2 pairs of chromosomes. And this is the 2N that you see right here. And so the 2 adults now want to have a baby, and so they produce the sperm and the egg, okay? And the sperm and the egg, each of them has N chromosomes. Only 23 in the case of humans, only 1 pair of chromosomes. Why? Because the sperm and the egg have to come together to form the fertilized egg. And in the fertilized egg, we get N from the sperm and N from the egg to come again to the situation where we get 2N, where we get again a diploid cell. And so this process of the creation of the sperm in males or the egg in females is called meiosis. And it's a very interesting process. And so in the process of meiosis, what happens is that I need to take a cell that has 2 pairs of chromosomes, 48 chromosomes, and make it into a cell, into a sperm or an egg that only has 1 pair of chromosomes. So how do I do that? This is just a diagram, and you can see the 2 pairs of chromosomes. So as this is just depicting, it's not depicting all 23 pairs, but let's say that 1 copy of chromosomes from the father is blue, and 1 copy of chromosomes from the mother is yellow, for example. And so I have 2 pairs of chromosomes in this cell. And what happens next is that the cell multiplies the number of chromosomes. So instead of 22 pairs, I have 48 pairs of chromosomes in the cell, so doubling the amount of chromosomes. And what happens next is a very interesting process called recombination, which is the main process that is responsible for diversity amongst human beings. And this is the main reason that actually a kid that is born does not look exactly like its mother or exactly like its father or exactly like a mix, but also shows some new characteristics maybe. And in this process of recombination, just little segments from the chromosomes jump to one another and are switched. And this is called recombination. What happens next is that the cell splits from 1 cell into 2 cells. And now each cell has 23 pairs of chromosomes. However, these chromosomes, because of their recombination, are a little bit different than what we started with. And so now, to create the sperm or the egg, these cells split again to create 4 haploid cells, meaning 4 cells that have only 1 pair of chromosomes. Yes?
Yeah, I was going to say, when you start out with an X and a Y, can you at some point end up with 2 X's? Or
what do you mean an X and a Y?
I'm sorry, chromosomes were categorized as X cells or Y in the last slide, right? There's X chromosomes.
I will explain about it later. Basically, the humans have 22 regular chromosomes, and then there's this additional chromosome called X and Y. These are the sex chromosomes, which are different for males and females. So females have 2 X, 2 copies of this, and males have 2 copies, 1 copy of X and 1 copy of Y. But these are just multiplied the same as anything else, so not to be confused with them. And so now we've created these 4 haploid cells, and these cells are actually the sperm and eggs. And so we have now cells that are ready to meet, because we have 1 N, 1 N, 1 N from the father, 1 N from the mother, and now they meet. And they meet to form this fertilized egg, which is also called a zygote. And the fertilized egg has, we are back to the situation where we have 2 N again. And now this fertilized egg needs to make somehow a baby, right? Because from 1 egg we need to come to the situation where we get this baby.
So you've got the 4 haploid cells. Do all 4 go into the egg and all 4 go into the sperm, or do they sometimes?
So these are just 4 sperms or 4 eggs.
Okay. So whichever egg
Whichever
and
is
sperm
first.
combine will dictate which
Exactly,
of
so
those.
only one of these as a sperm and one of these as an egg will come together.
Yeah, the initial diploid cell, is that the sperm or the egg or what?
Initial diploid cell,
The initial cell that you started with.
which one? This?
The one on the very left.
Oh,
This one. Yes, it's a diploid cell. So I start with a regular cell. All cells in the
And
body
that's
are
part of the
normally. Sorry?
That's part of the egg, or it could be anything.
It's a cell that is intended, in this case, to become the egg or the sperm. Yes, but it starts out as a diploid cell. And so I come to the situation again when I have the fertilized egg. I have 2 N again. All is good. We are haploid again. Diploid again. We have 2 times N chromosomes. And now this one cell needs to split again and again and again and to divide itself until we create a baby. And so we need a lot of cells to make a baby. So the cells needs to divide again and again and again. And this process of division, of regular division, is called mitosis. And so in mitosis, I start with a diploid cell. I start with a cell with 2 N chromosomes. And I end up with 2 cells that are diploid, 2 cells that are 2 N chromosomes. Simply, I just take one cell and I split it into 2 while making sure that the number of chromosomes are left intact, that I still end up with 2 N chromosomes in each cell. And so in this process, just simply, so one set of chromosomes is the blue from the father and the yellow from the mother, and the chromosomes simply multiply themselves. After they multiply themselves, the cell splits into 2. One goes into one cell, and the other set of chromosomes goes into the other. Any questions? And then the baby grows up, of course, and the circle of life begins over and over, and this is why it's called a circle of life. Any questions about this?
What goes wrong? So
My focus on the wrong.
That's usually,
What do you mean what goes wrong?
in genetic diseases, where did bad things
So-
happen where you're missing a chromosome or?
So by genetic diseases, you mean diseases that are inherited from
Yes.
parents. And so there are 2 types of diseases that are called genetic. Ones are just genetic diseases that can be acquired throughout a lifetime and are not inherited, and parts that are inherited. And so if they're inherited, it's simply, the chromosomes that are passed through the sperm or the egg already contain these mutations from the parents, and then they're just simply passed on.
But aren't there cases where there's like multiple X or too many X?
Yes, there are cases,
So what
for example, Down syndrome is caused by something that is called trisomy 21. So instead of 2 copies of chromosomes, that we get from the mother and one from the father, somehow in the process of replication of the chromosomes, there are just created 3 chromosomes of 21. And this can cause mental retardation. And there is also trisomy. Trisomy just means 3 chromosomes instead of 2. And there is also trisomy 13 and trisomy 18, I think. And yeah, so there are cases like this. And
But
so,
they don't know where in the process that happens?
I'm sure they do. I'm not sure where exactly in the process. I'm pretty sure it's somewhere during the DNA replication. So something is replicated additional times. Yes?
Okay, so sorry, I've just always been curious about this. I never found out because I think they have always used pea plants in the past.
Did they have what done?
Always used pea plants for the example in the past, so it's never come up. But if you go back one slide, so you have the X and the Y
Mm-hmm.
from the father and the. At what point does the gender get decided, right? Because why is that Y not getting passed through the whole process?
So I will be glad to answer that after class.
Okay.
I will not go into that now, but come to me later and we'll talk about it.
Okay.
Okay? So yeah, I'm sorry. I know everything is interesting. I couldn't shove everything into the presentation, but please, I didn't say it in the beginning. If you have any questions, feel free to come to me today or tomorrow and ask me anything. And I can also leave you my email if you want to ask anything. And also, you can search it online. Most of it is available. And so next, I want to talk. So we talked about DNA and how it has to do with inheritance and characteristics. And now I want to talk a little bit more about what exactly is DNA and dive into more into the subject and what is the structure of DNA. But before, a little bit of history. So I think it's always important to start, with all the experiments way into understanding the DNA. And so before 1928, people had some idea about characteristics being passed on, but people didn't really know what was responsible, or what was the material responsible for passing on. So the word DNA was not really known in 1928. However, a breakthrough experiment that happened in Griffith's lab started paving the way into understanding this. And what Griffith did, so Griffith's lab worked with bacteria. And in the experiment of Griffith's lab, what he did is he worked with this very aggressive type of bacteria. It's shown in pink here. And this bacteria, if you took it as is and injected it into a mouse, the mouse died. The mouse could not handle this aggressive bacteria and caused the mouse to die. What Griffith did is he heated up the bacteria in the speaker, and he caused it to be killed. And so in the speaker, there's a heat-killed bacteria of the aggressive type. And when he injected the heat-killed bacteria into the mouse survived. So heat-killed bacteria did not kill the mouse because the bacteria was dead. What he did next is the interesting part, is he took another strain of this bacteria, and this bacteria is the non-aggressive bacteria. It's shown here in blue. And so he took a living non-aggressive bacteria and mixed it with the dead heat-killed aggressive bacteria. And so in the speaker, we have a mixture of the living non-aggressive type and the dead aggressive type of bacteria. And he took this mixture and injected it into the poor mouse. And the poor mouse, of course, died. And surprisingly, when he isolated the cause of death from the mouse, he found he could isolate living strain of the aggressive bacteria. So that was very surprising. And when he thought about it, he concluded that in the speaker, something was transformed from the dead heat-killed bacteria into the live non-aggressive bacteria. The non-aggressive bacteria gained characteristics of aggressive bacteria. And those characteristics of the aggressive bacteria actually killed the mouse. And so he called this process transformation. And indeed, this process is known today that bacteria can transform genetic material from one to another. Even if we kill the bacteria, still the DNA or the genetic material of the bacteria stays in the beaker. And so it can be transformed, and this process is called transformation. However, this was in 1928, and only in 1944, Avery, McCarthy, and McLeod, in a very breakthrough study, have shown that DNA is the material that was transformed in that beaker. DNA was responsible for the transformation of these characteristics of the aggressive bacteria to the non-aggressive bacteria. And only in 1953, Watson, Crick, and also Rosalind Franklin, that is not always mentioned, discovered the molecular structure of DNA, which led to more experiments and more discoveries regarding what exactly does DNA do and what processes exactly does DNA participate in. So what did they discover? What is the structure of DNA? So DNA is a double helix. Double because it has two strands that are one opposite of another. And helix because it has this coiled shape that looks like a helix. And DNA is built, and this I'm sure you know, of a four-letter alphabet, A, C, G, and T. And these are the molecular structures of these A, C, G, and T. So A stands for adenine, G is guanine, T is thymine, and C is cytosine. So these are just molecules or molecular material that are the alphabet of the DNA, and the DNA has 4 times 10 to the 9th. The human DNA has 4 times 10 to the 9th nucleotides. And on these two DNA strands, A is always opposite of T. So you can see it also here. So if I have A on one strand, I will always have T on the other strand. And C is always opposite of G. So if I have C on one strand, I will always have G on the opposite strand. And they're also bound together by some ties that hold these two strands together. And so DNA stands for simply a word that describes the molecular structure of the DNA, deoxyribonucleic acid. And this is simply saying that the DNA is composed of 3 main things. And the first one is a sugar backbone. So the sugar here, you can see it in orange, it's the orange square here. And you can also see it here, you can see its molecular structure. These black dots are carbons. Carbons are usually numbered from 1, and you can see there are 5 carbons here, from 1 to 5. And so this is the sugar backbone. And the second material that composes these nucleotides is a phosphate. And the phosphate, you can see that it's attached to the 5th carbon. And also, the nucleotide is the base. And the base can be either A, C, G, or T, simply these molecular structures that are attached to the first carbon, and the base determines the type of nucleotide. So I can have a total of 4 different nucleotides. And so we mentioned before that DNA doesn't look just like a long strand. However, if we did take the DNA, connect it together, and stretched it, the human DNA, we would get something equivalent to 1 meter long. Just so you know this, we said that each cell contains the DNA. The nucleus of each cell contains the DNA. However, the size of a human cell is roughly 5 micron. So how do you get 1 meter into 5 micron? You have to make the shape of the DNA very compact, and you have to pack the DNA in a certain way so it'll fit into these 5 microns. So this is exactly what happens. The DNA here is this pink stripe, these pink stripes. And you can see that the DNA is actually coiled around this core and then set into this circular shape. And then these circular shapes are actually packed on top of one another. So this is actually the DNA, this pink thing. And they're packed on top of one another, and then this structure is then coiled again and again and again and again until it's very packed, and until we get this familiar shape that we saw before, which is the chromosome. And this is why I said when I say DNA and chromosomes actually mean the same thing, because chromosome is just the way that the DNA is packed. It's just the way you can see these chromosomes. Any questions? Yeah.
So the chromosomes they're disconnected from each other? It's not one big chain?
Yeah, they're disconnected. It's not one long chain that you can stretch. Humans have 23 chromosomes, or 23 pairs of chromosomes. So they're just different parts.
And each cell has one exact copy of all of in the nucleus?
Yes. So all the cells have exactly the same copy of DNA in the nucleus. Yes.
It doesn't matter if it's a blood cell or a skin cell.
Yes, some cells do not have. Some cells just don't have nucleus, so they don't have the genetic material, such as blood cells, for example.
Those are the impression that chromosome formation happens only around the time you're going to undergo meiosis.
Yeah, yeah. So basically, I know, I haven't been very honest with you. This shape that you see here, I was trying to keep it simple, but since you asked, this shape here is actually the only thing that we can see through a microscope, because it's very packed and it's very large. However, this shape here is only happen through the time that the chromosomes multiply themselves. So what we see here are actually, so in regular situation where the cell doesn't divide, our chromosomes actually look like one of these arms, only one of them. And when we said during mitosis, the chromosomes multiply themselves, right? Because we need to make enough chromosomes to have enough chromosomes for 2 cells. And so when they multiply themselves, they look like this. So this is one copy, and this is the other copy of the same chromosomes. They're identical to another, and they're actually connected through this piece here. But they are later on separated to fit into the 2 cells. So this is actually a chromosome that is, and when there are only one arm, when the cell is not dividing, you can't really see these chromosomes through the naked eye, or of course, or through a microscope even. Only when the cell divides and the chromosomes multiply themselves, then the shape of the chromosome is big enough to see through a microscope. And this is why most shapes of the chromosomes that you will see through electronic microscopes and such will look like this, but this is actually 2 copies of the same chromosomes because it's when the cell divides.
So does the folding change from species to species?
That's sorry?
The folding of the DNA, does it change from species to species?
No, DNA is packed. I mean, the control of the packing might be different. So the control mechanisms differ among different species, and they're highest for human beings because they're most complicated. Most complex. But the main shape, the chromosomal shape, is more or less the same.
Is there any significance to like where in the chromosome it decides to split sort of horizontally, like where the center of the X happens?
This?
Yeah.
So this is just, what do you mean if there is a change?
No, I was wondering if there's any significance to it.
To why it's connected here?
Yeah, it's just clearly like different chromosomes split at different points. Is that point?
I'm actually not sure that it's exactly the same point for each and every chromosome. I think it might be a different point. I don't know why. Yeah. I'm sure there is significance, by the way. There's significance for everything in biology. Yeah.
So you said there are 22 pairs of chromosomes. So so one chromosome is one strand, is it?
Mhm.
And there are like 44 strands essentially.
There are 23 pairs of one strands. So think of the map that I showed you before, without this shape, but just one of these. And there are 23 pairs of these. And only when the cell wants to multiply itself, then these two pairs are, these one strand is multiplied into two. One goes to one cell, because one cell divides into two. One goes to one cell, and the other goes to another. Yes?
And you're saying that normally that they don't exist in pairs like shown here. Normally it's just a single arm.
Yes.
But every time I ever see chromosome illustrated, they're always illustrated in pairs.
Yes, and I explained it, it's because
Because that's what we can see.
we can't see it. Right. So if you want to see the chromosomes, you usually catch the cell when it's dividing, and then before it divides, when the chromosomes are doubled up in the cell, and they're more packed and more have this shape that can actually be seen from the microscope. So next we'll talk about something that is called the central dogma of molecular biology, and this is it's called the central dogma because it involves DNA, which is the most important material that a cell can have, because DNA is actually, you can think of it as the book of instructions on how to make an organism. And you can flip in the pages, and each page can have a different characteristic or instructions for something different that can be done within this organism, within this living cell or this living thing. And this book of instructions can actually go through different processes. So DNA can go through different processes, and there are three main processes that DNA goes through, and it actually flows from one process to another, and the normal flow of this biological information of this DNA is called the central dogma of molecular biology. And so we're going to learn a lot about each process, a little bit about replication, but more about transcription, and tomorrow we'll go in detail into translation. So what are each of these processes in short, because we're going to talk about them more. So replication, we saw. So during cell division, DNA has to multiply itself, so during what we call mitosis. And so DNA actually flows into DNA. So DNA creates a copy of itself and then divides between the two daughter cells. Transcription is when I want to take this DNA and make a copy of it. And the copy is RNA. And so why would I want to make a copy of the DNA? So I don't actually make a copy of the whole DNA. That's here, the replication. What I want to make a copy is a certain page in my book. So I want to read the page that tells me what is the shape of the seed of the pea plant. So I flip into my book of instructions on how to make a pea plant, into my book of DNA. I land into this page that explains how to make the shape of the seed of the pea plant. And this page actually describes the characteristic of the pea plant. And now I can tell you that this page is actually the gene. So a gene is actually sort of like how we define allele. It's a segment in the DNA. It's a page in my book that describe a certain characteristic that I want to make. And so why do I want to make a copy of this gene? It's because if I want to reference my book in a certain page, I don't want to go to the library, take my book, rip out the page, and go and read it somewhere else. What I usually do is I take this book and I take it to the copy machine, I make a copy of that page, because I might want to refer to that book later, or somebody else will want to refer to it later, and I don't want to just rip up that page. I want to make a copy of it and use my copy. And so this is exactly what happens in transcription. DNA is transformed into a copy of a specific gene, into a slightly different material that is called RNA, and this gene is then being transcribed into a process that's called transcription. What happens in translation is, okay, I read the book of instructions, I read my page saying how to make the shape of the seed round, for example, and now I want to go and make that shape of the seed round. I want to do something with all the instructions that are read from this gene. And the structure in the cell that actually gets things done is a protein. And through the process of translation, my copy of the gene, my RNA, is being translated into a protein. And the reason it's called translation is because if I deal with DNA and RNA, which is very similar to it, I have a 4-letter alphabet, A, C, G, and T. And a protein actually has a completely different alphabet. It looks completely different than RNA or DNA. It has an alphabet that is composed of amino acids. And so in order to translate my 4-letter alphabet into my 20-letter amino acid alphabet, I need to translate. I need to use something that is called translation, and we'll learn all about this tomorrow. Any questions? Okay, so let's talk a little bit about replication. And so during DNA replication, we already talked about this, DNA is precisely replicated during cell division. And I don't think I have emphasized the word precisely enough, because this is very important. I have one cell with my DNA, and now I want to create two cells. But I need to make sure that my copies are precise and are accurate into these two daughter cells. I don't want to have a different book of instructions in every different cell. I don't want to have any mutations or anything that is written wrong in my book of instructions. I want to have a very precise replication of my DNA. And so the way that we make sure that the DNA replication is precise is through something that is called a semi-conservative replication. And so the semi-conservative replication takes advantage of the fact that the DNA is double-stranded and that I can use one of the strands as a template for the other strand. Because what do I know of my DNA? If I see an A on one strand, what do I see on the other strand? What do I know? Which letter is always on the other strand? A is always opposite of T, right? And if I see C on one strand, G. And it's not that complicated. And so I have two DNA strands. Orange will, so both of them serve as templates. So orange here is a template. I split the DNA up. And so the top strand serves as a template to a completely new DNA strand. And the bottom strand also serves as a template for a completely new DNA strand. And so this is why it's semi-conservative, because I keep part of my old DNA and then I put a new strand of DNA on top of it. And so from one copy of DNA, I end up with doubling my DNA content with getting two copies. And so the structure in the cell, the enzyme in the cell that is responsible for all of this is called DNA polymerase. DNA because it deals with DNA, and polymerase is because it catalyzes the polymerization, building, of nucleotides into new DNA. And so DNA polymerase is actually responsible for looking at one strand, seeing a T, taking an A, and putting it in front of it, and therefore building the DNA. And however, there are always problems. It can't just come to a strand and start throwing nucleotides on it. It needs to start from somewhere. It needs something that is called a primer. So primer is simply a small DNA strand. It's 5 to 6 nucleotides long, and it's simply the start. And so DNA can elongate this primer. It sees this red nucleotide, and it knows to add this blue nucleotide in front of it. And so the accuracy of the DNA polymerase is pretty amazing. It's 1 parent mistake in 10 to the 9th nucleotides. And considering our DNA is 4 times 10 to the 9th long, then it doesn't normally make a lot of mistakes. And there are also additional proofreading mechanisms by additional structures in the cell that if by mistake there was a pairing off the wrong nucleotide, there comes another structure in the cell that removes these nucleotides and allows the DNA polymerase to continue this polymerization. And so just because I'm not going to go into the details of the DNA replication, I just wanted to show you this little structure, just to, as a lesson that nothing in biology is simple. And so basically, the DNA polymerase, which is just this little structure here that we talked about, is one of many protein and many structures in the cell that participate in this amazing mechanism that is called DNA replication. And so these additional players are many. We have the helicase that actually knows how to separate the DNA strands before replication, because someone needs to pull these strands apart. If we have the parental DNA double helix here, then it's pulled into 2, and each strand is replicated through this semi-conservative replication. We also have the primase, which is the enzyme that is responsible for putting on this primer, this beginning for the DNA polymerase, on one of the strands, or both. We have additional proteins that are called single-strand binding proteins. And they actually do what they sound like. They're right here. So when the DNA is separated and I have one strand and another strand here, they actually keep the DNA from premature annealing, because DNA has this characteristic that A always sticks to T and G always sticks to G, and it can start folding and sticking to each other, and so it simply keeps it intact and in order. Topoisomerase is a very exciting protein that actually can regulate DNA over or under winding. So DNA is a double helix, the DNA and turning it, making sure that the DNA doesn't over or underwind, and making sure that everything goes smoothly. And there are many more additional proteins that I haven't described, and just to give you an idea of what is DNA replication in short. Any questions? Okay, so next, and this is our last topic for today, I'm going to talk about transcription. So during transcription, we want to copy a specific segment of the DNA, which we now define as gene. So in our book of instructions, we want to open this our book into one page, and this page gives me a description of how to make something in the body. For example, what will be the color of the eyes of the newborn baby. And so this page, or this segment in the DNA is called a gene. And we want to make a copy because we want to go and read it and do something with it later. We don't want to hurt our full book, our DNA book. And this copy is actually in the form of RNA. So what exactly is RNA? RNA stands for ribonucleic acid. And here you can see a picture of the DNA and the RNA. And so if the DNA, we know it's a double helix. And it has a four-letter nucleotide, four-letter language, A, C, G, and T. And so the RNA is, first of all, it has a slightly different structure than DNA. And also, we can see that the four letters of the RNA are A, U, C, and G. So instead of T, instead of thymine, I have uracil. I have U. So this is slightly different, but still very similar. One thing that we can clearly see is that it has only one strand and not two strands. And the fact that RNA is only one strand actually allows it to use the characteristic of DNA or RNA, which is sticking to one another. So in this case, A always sticks to U, C sticks to G, and then so this RNA strand can stick to each other and make three-dimensional structures such as this one. And most times anything in the cell with these three-dimensional structures can actually go and do things. So RNA that actually sticks to one another can go and perform catalytic functions sort of like proteins. And we'll learn all about proteins and what they do tomorrow. However, the RNA that I'm talking about here, the RNA that serves as a copy of our gene, does not do this. It serves for a completely different purpose. So what happens is that during transcription, so I want to make a copy of a gene. I received the decision I want to make a copy of a certain gene. So during transcription, the DNA opens up at a specific place, which is the gene. And one of the DNA strands serves as a template, sort of like DNA replication, for the synthesis, for the creation of the messenger RNA. And I call it messenger RNA because it carries the message of the gene. And so if this is my DNA, these are my two strands of DNA. DNA opens up, and one of the strands serves as a template to create this messenger RNA. And the structure in the cell that creates this messenger RNA is RNA polymerase. So this name is familiar. RNA because we're creating RNA from DNA. So this is a slightly new material. And polymerase because it performs polymerization of this RNA strand. And it differs from DNA polymerase. So just to remind, DNA polymerase was the enzyme that actually catalyzed the polymerization of DNA during cell division, during DNA replication. But RNA polymerase differs in, first of all, it doesn't need a primer. It uses U instead of T because it creates RNA, not DNA. And the accuracy is, it's less accurate than DNA polymerase. It makes one pairing mistake in 10 to the 4th nucleotides. And also the proofreading is not as elaborate as the DNA polymerase. It performs the proofreading by itself. And the reasons are two. Anybody can think of why the RNA polymerase is less sophisticated than the DNA polymerase? Yeah. Not necessarily.
Cheaper. Maybe because if it doesn't work, then the cell is not performing its function, but it might be able to continue reproducing.
Okay, close. So first of all, the length of the messenger RNA is much shorter than the length of the whole DNA. So a gene, and you couldn't have known this, but a gene is only a couple of thousand nucleotides long, and we said that the whole human DNA, so during DNA replication, we need to replicate 10 to the 9th nucleotides. So it's much shorter, so we can allow more mistakes. And second, don't forget this is only a copy. So our book remains intact. We went to the copying machine, we made a copy. So if we make one mistake here, I mean, it couldn't be good, but it's also not as bad as hurting the book of instructions, hurting the DNA itself. And so we can allow more mistakes here.
Does the fact that it's shorter mean that you're less likely to make a mistake than a longer replication?
It has to do just with the accuracy of whoever makes this replication, whoever makes this transcription. And here you might be more likely to make a mistake compared to the DNA polymerase, but it's also shorter. So the chance that you'll make a mistake is only 1 in 10 to the 4.
But you said that the length of these are on the order of 10 to the 4, you said?
They're on the order of thousands to a couple of thousands.
So it seems like the error rate is actually about the same, like as far as.
The error rate could be the same.
Yeah, in terms of like frame.
How long they are, yeah.
Yeah.
If you refer to it might be the same, however, even though the proofreading is done here by the polymerase itself, so you do have less control over making mistakes.
Yeah.
So just showing that the cell has more control over the replication, or wants to control more of the replication, make sure that there are less mistakes there. Because during replication, even if you make a mistake, then it could be repaired by some other mechanism.
What is the proofreading for this and for the DNA? What exactly?
The DNA polymerase, so for the DNA polymerase, proofreading is done by a completely different structure in the cell. An enzyme called exonuclease, because it makes the wrong nucleotide exit from.
So does error correction, some type of error correction?
It's exactly error correction in the DNA polymerase. But here, it's not. Here, the RNA polymerase itself makes the proofreading. And part of the mechanisms of proofreading are actually, so the DNA polymerase, part of the proofreading that it does for itself is that it has a higher affinity for nucleotides that are paired correctly. So if it stands in front of an A, it has higher affinity for T's. So I imagine that this is sort of the same. So RNA polymerase doesn't have any outside helpers, but it has higher affinity for correctly paired nucleotides.
There's probably some robustness in the way proteins are created from the messenger RNA, right?
So let's talk about proteins tomorrow.
Yeah, but the question was,
Okay.
shouldn't the right metric actually be what fraction of proteins come out wrong? So is that much smaller than 1 in 10 to the 4, or is that of the same order?
So you're talking about errors in translation.
Yeah, after you do the translation, is the frequency of errors a lot smaller than 1 in 10 to the 4, or is it of the same order?
You can't measure it that way because it's a different language. So the errors in the proteins are made in the language of the proteins, that is amino acids and not nucleotides.
You could ask how many proteins come out wrong, for instance, from what was intended as a fraction, right?
So if a mistake happens here, so I will just explain what happens next with this messenger RNA, and we'll learn this tomorrow, is that it's translated into a protein, which is something completely different, a different structure that actually goes and does the job. And if there's a mistake in the messenger RNA, if there's a pairing mistake, if there's a mutation in one of these nucleotides, then this will be translated on to the protein, and the protein we created will be wrong.
Yes, I was just wondering, I don't know the answer. The question is whether there's a robustness in the translation of messenger RNA to proteins. It sort of makes up for these errors.
There is redundancy in the code, and basically, usually you have several triples going for the same amino acid. In the last symbol, you can change it pretty much for a lot of these codings arbitrarily, and you still will get the same.
Hopefully I'll have time to, and I can actually show this to you in my, I did want to talk about this tomorrow, I'm not sure I will have enough time, but regarding what exactly is translated into a protein, I will talk about tomorrow, and then we can talk about this too. But I think I understand your question now.
Question? So
Yes.
also, what do you mean by DNA replication error? Chemically, it's not possible, for example, for A to bind to anything other than T. So by an error, you mean like a deletion of an actual pair, or?
So I haven't talked about deletions, but when you build the DNA on top of a new strand, if you see an A, then the natural choice would be a T, to put a T in front of it. However, so a mispair would be that the T is put in front of it by mistake.
So that's chemically possible.
So it is possible. It happens. I mean, sometimes all the proofreading mechanisms and they just don't work, or they miss one nucleotide. So it does happen.
Sorry. How do we find the genes?
How do we? Sorry.
How do we know when the gene starts?
Okay, so how does this mechanism know when the genes start? So it actually has it actually has sort of something that marks the beginning of the gene and marks the end of the gene. So it marks, I didn't read it here, but it marks for the RNA polymerase where to start and where to end.
So it's an additional structure other than the base.
It's not an additional structure, but it's the way the nucleotides are ordered within this DNA. This area, so imagine this is A, C, C,
Oh, I
C,
see.
for example. So if the RNA polymerase sees a certain sequence, it knows that it's supposed to start from the sequence.
So but it means it needs to read the whole DNA and clearly find the sequence.
So there are additional players in here that actually know exactly where to go in the DNA, which chromosome to approach, and which part of the DNA to approach. So it's not just the RNA polymerase itself, but as we saw before with the replication, it's not only the DNA polymerase, but additional players are in this process of replication, and the same goes here with the transcription. So I'm not bringing you the full story due to lack of time, but just so you know, so any of the processes that I talk about, it's not just one, it doesn't happen just once, or it doesn't just happen by one enzyme. It's usually a bunch of enzymes working together to make this process happen.
In each chromosome, is the sequence of the letters fixed, or is it fixed over time, or does it vary, or is it
So in
fixed?
each individual, it's fixed.
It's fixed.
Yes.
So whatever he was talking about DNA sequencing, it's sequence of those
Exactly.
letters for all chromosomes?
Exactly. So full human genome sequences actually aims to sequence the whole human genome.
So it's like, it's the 1 meter long?
It's the 1 meter long, the 4 times 10 to the 9th nucleotides. And this is the reason why they have to split it up into little segments, because it's not accurate or almost impossible to read such a long sequence. And so lastly, I want to talk about a process, a very interesting process that is called alternative splicing. And so alternative splicing, this process itself, was discovered in 1977, which is fairly recently. And it was discovered to occur only in eukaryotes. And eukaryotes are those multicellular organisms with a lot of cells, such as humans. And when the human genome was sequenced and started to be analyzed, and the actual number of genes in humans started to be clear, they discovered that actually humans have around 25,000 to 30,000 genes. Initially, people thought that humans have around 2 million genes, because look at us, we're so complex, and there are so many things that need to be done inside the cells, and to create a human being, there must be tons of genes, and each gene is responsible for something that occurs within our cells. And to look at this number, this is a fairly small number, just to compare it to other species that we know of, for example, rice. Okay, look at us and look at rice. Rice has more genes. It has 37,500 genes. The dog, with all due respect, has 19,000 genes. And even more insulting, C. elegans, which is an earthworm, it's a very highly studied earthworm, has 20,000 genes. I mean, we have 25,000, and the C. elegans has 20,000. And look at the differences between us and the earthworm. You can see that we operate a bit differently. So how come? What happens there? How come we have that little genes? And so then alternative splicing, when it was found out, it sort of explained this phenomena that actually the gene includes not this one sequence, but we can actually characterize it into 2. So part of the gene are exons. These are the brown parts. Exon stands for express sequences, which are the coding sequences. These are the sequences that actually end up in the finished messenger RNA and that are translated later on to a protein. But it also has some introns. Introns are these blue parts, and they are intervening sequences, which are later on taken from the messenger RNA. Once they were called junk DNA because nobody really knew what they were doing. They were called junk because it's very easy to categorize something like junk. But now we know that they actually have some sort of control mechanisms that control the creation of the RNA and the gene. And for example, some of the introns may serve as the starting point of this gene and the ending point of this gene, for example. And so we can see that if the DNA is actually composed of these exons and introns, and only the exons, only the brown parts, end up being in the final messenger RNA. And so why does this explain the low number of genes in the human chromosome and in humans? And so different combinations of these exons can be used, different combinations in one gene can be used to create different messenger RNAs. And different messengers messenger RNA. From this gene, we have 5 different variations of messenger RNA. And later on, it was found that 95% of the genes in the human genome actually undergo alternative splicing. And so, due to the fact that we have that little amount of genes, each gene can actually make multiple proteins.
So is it no, it doesn't happen in rice?
It does happen in rice. So rice is a eukaryote. It's a multicellular organism. However, the extent of maybe it is more complex than us. Maybe we'll find something that we don't know. Research is not done yet. So yes.
So if the gene here is not a continuous string of only exons, now how can we decide or define when a gene starts?
When a gene is what?
When a gene begins in downstream DNA.
How can we define the beginning, the start of the gene? So this was asked before. So part of these introns, part of these control sequences that we say, actually mark for the RNA polymerase where to start transcribing. And we also have part of the DNA that is sort of a mark for the RNA polymerase where to stop transcribing. So it actually marks where is the beginning and where is the end of the gene. And it's not only the RNA polymerase, but it's additional players that take part in this whole process that know how to approach the correct chromosome, open it up in the correct place. So that concludes our first section of this tutorial. Tomorrow we'll learn more about proteins. So mainly we'll start with the translation process, and no worries, I will remind you what we learned here today in short. And also talk about some of the processes that these proteins participate in, such as signaling network, and we'll also give some examples.
Question.
Oh, we have one
Yes.
question.
I wanted to ask you about the size of the alphabet. Why 4?
The size of
the alphabet? Why 4? I mean, there are good reasons why it should be an even number.
Please do.
This reminds me a lot of what was discussed here today. What is the minimum number of possibilities that can make up information? So yeah, I don't know exactly why 4. I guess I can always say evolution.
But is there any evidence of quote unquote DNA that use different alphabet than a size 4?
No. So the only thing that is known about DNA, the only DNA that was found until today had 4 letters, A, C, G, and T, and with a variant of RNA that has U instead of T, but no others.
So there is no conjecture why that is kind of like a good number, like an optimum number, as opposed to 2 or 6 or 8?
I'm sure that some papers discuss about this, especially papers that deal with evolution. But I guess you can ask this about a lot of things in life, why transcription, why replication. I can always say evolution, but that would be too general, and yeah.
So all organisms use those ACTG, all of them?
Yes, all of them. And moreover, all organisms use the language of 20 amino acids to create proteins, and so no other amino acids besides these 20 amino acids was ever discovered, at least not on planet Earth.
Is the artificial DNA created in the lab at different times? It's bases now already?